I am Claes Magnusson, and this is an introductory tutorial about the Baxter algorithms, a software package for automated analysis of microscope image sequences, and in particular for cell tracking. The software is written in MATLAB, and you can either run the source code in MATLAB, or you can run a deployed version of the program which doesn't require MATLAB or a MATLAB license. This is the main window of the program, and to open an experiment I press File, Open Experiment, and now I'll choose a bright field dataset with muscle stem cells. Here's the image sequence of the experiment, and here is the first image of that image sequence. And all of the operations that I can perform on this image sequence can be found in these menus. The menus have different levels. And now I have the basic level, and that means that only the basic functions will be shown. And these are the most important functions that you will use most often. So for example, there's only open experiment here. And if I select advanced, I get more options and more flexibility, but I also get more functions to keep track of. So to start with, we will have the basic level. And the development level is only for functions that are under development or which have very limited use. So don't use those. If you need help with anything, or if you don't understand how to use a function, you can click Help and User Guide, and then you get a PDF with detailed instructions. So to start with, we will play the image sequence, and then you can visualize the image sequence over time, and you can also zoom in to different regions to look at them in more detail. It's good to look at the image sequence before you process it to make sure that everything is the way you want it to be. And then in order to track the cells in the image sequence, we need to find the outlines of the cells and then link them into tracks. And finding the outlines, that step is called segmentation. And there's a specific user interface where you can set parameters for that step. So we click on Set Segmentation Parameters, and then we press Update to see what the current segmentation looks like. And to improve the results, we can change the segmentation parameters. For example, these regions are not really cells, so we can increase the minimum area to, let's say, 500 pixels to get rid of them. Or that wasn't enough, so maybe 1000 pixels. That removed that one as well. And then we might think that this region is too big, and then by increasing this threshold we can make it smaller. And once we're happy with our segmentation, we should of course look at multiple images to make sure that everything works. And it seems like the minimum area was set too low, so maybe 500. Yeah, then we don't remove this one. We should actually not make it too small. But once we're happy with the segmentation, we can go to the track linking step. And that's done here under Automated Track. I won't go through this now, but once we're done with the tracking step, we can go to manual track correction and look at the tracking results. And now I'll just open some old tracking results. We can play the tracking results. These white things are objects that were found by the segmentation algorithms, but which weren't included in any of the tracks. And we can click in the lineage tree to move to different time points, and we can change the assignments made by the track linking algorithm by choosing this tool and clicking on the cells. Now this is obviously wrong, so I'll correct it back. And we can also change the outlines of the cells that were computed in the segmentation step. So this one is obviously a little bit off. So I'll select the zoom tool first, and then I'll select the outline editing tool, and I carefully outline the contour of the cell. This is of course not necessary if you're only interested in the migration velocity or something like that, but for some applications it might be necessary. And once you're done validating your results or correcting them, you can go to the analysis menu, and here's cell analysis GUI, which lets you look at parameters for each of these cells. So here's the axis ratio, and it increases when cell stretches out. And we can look at the cell size, and we can see that the cells start out small and then they grow until they divide and give rise to new cells. We can also look at the total distance traveled by every cell. You can see that most of the cells have the same velocity, and that the cells are slightly slower in the beginning of their life, or when they've just been created through mitosis. And this can be done both for transmission microscopy and for fluorescence microscopy. And to show that it works for transmission or for fluorescence microscopy, I will open a data set from the cell tracking challenge, the ISBI cell tracking challenge. These are mesenchymal stem cells where the cytoplasm is fluorescent. And the tracking results for these cells look like this. You can switch between the image sequences by pressing previous and next. And the tracking works both in 2D and 3D if you have fluorescent images, and you can have fluorescent cytoplasm or fluorescent nuclei. To show that, I will open a C. elegans dataset where the nuclei are fluorescent. So this dataset is also from the cell tracking challenge. And in the track correction user interface, we can look at the tracks from above. This is a maximum intensity projection, and we can also look at other maximum intensity projections, or we can look at 3 projections at once. They're seen from above and seen from the two sides. And we also have the lineage tree, and we can go to a time point by clicking in the lineage tree, and we can select cells and identify which cell is which in the 3 different views and in the lineage tree. So these were the different data sets that I was going to show for tracking. And in addition to tracking, you can analyze fluorescence or histology sections with fluorescent proteins. And it's been designed for analysis of muscle fiber histology. Here's a data set where we've injected GFP positive cells into a muscle without GFP. This is mouse muscle. And then we can see which fibers were generated from the transplanted cells. I'll just choose one of them and show you how the analysis works. You press Fiber Analysis GUI, and this assumes that you've already segmented the cells or used the tracking module to generate segmentation results. So I will only do the show the merged images with the outlines of the positive fibers. And here I select the tracking version. I select the GFP channel because that's what I want to analyze. I choose the median inside each fiber instead of the mean, because we have some grains of secondary antibody that would skew the mean but not the median. And then I tell the program to perform automated thresholding to decide which fibers are positive and which are negative. Then I press start. And here is the fibers that the program found to be positive. We can also look in the terminal window where we have some output, and we see here that there were 183 positive fibers. We shouldn't close this window, because then we're closing the whole program. And I will end by showing analysis of myotube fusion. So here we have a data set where the nuclei are stained blue using DAPI staining, and the cytoplasm or the cytoskeleton is stained red using phalloidin. And then we go to the myotube fusion analysis user interface, and I'll only do this for one of the images. And now the program will find which regions are fused and which are unfused, and the fused regions are shown in white and the unfused regions are shown in gray. And the program also computes something called the fusion index. If you have all of the objects fused, it's going to be 1, and if you have no fusion, you have 0. but I'll explain this in more detail in the dedicated Fusion tutorial. And as I said, this tutorial was not meant to show how to do stuff, it's only meant to show what you can do. And then you can look in the other more detailed tutorials to find out how to do everything. Good luck!